Hello grade 12s. In this video we are going to start getting into the real sort of, meat and bones section of DNA in totality. So all the videos that I did previously to this were leading up to this section of protein synthesis. So make sure that you've gone through all the previous videos to make sure you have a solid understanding. And what I've done is I've separated this into a part 1 and part 2. So part 1, I'm going to go through a general overview. This is what protein synthesis is as an overview. And then in part 2, I'm going to go a lot more into the nitty-gritty. This is kind of like the questions will be asked. What will I need to be able to do for this protein synthesis section? So when I talk protein synthesis, what am I actually talking about? So as the name suggests, I am talking about how am I going to make proteins, synthesizing proteins. And why is that important for us? So go look in the mirror, go look at anyone else who's around you, anything else that's organic around you, and you will be looking at proteins, because proteins really are structure. So everything that gives us structure comes from proteins. And when we look at us as animals, and humans in particular, anything that is hormone or enzyme or immunity based all comes from a protein base. So having an understanding of this process is incredibly important for us. So just as a reminder again from previously what we discussed, proteins are made up of units called amino acids. So the monomers of proteins are amino acids. If I was to break down a protein into its most basic form, it would be an amino acid. And therefore, I need to link amino acids together to therefore make proteins. So in these videos, what I'm going to be doing is showing you very short chains of amino acids, which is going to represent a protein. However, in real life, these are really long chains of amino acids. There's lots of them that come together to form these really complex structures that are proteins. But as a rule of thumb, if you have a chain of less than 50 amino acids, you will be looking at a polypeptide chain. And a chain of more than 50 amino acids will be a protein. So make sure that you know the difference between those two. So as I said, amino acids join together to make proteins because they are the monomers. So in this little video series, I'm going to be using shapes to represent amino acids. So as we look here, I'm going to show you a chain of 4 amino acids. So I've used shapes, a square, an octagon, a triangle, and a circle, and each of these represents its own amino acid. So 4 different amino acids. What we also notice is that there are bonds that are holding these amino acids together within this protein structure. What are these bonds? Well, before we answer that, let's just have a reminder. I told you about DNA, that there are bonds that exist between the nucleotides of 2 adjacent polynucleotide chains, so between the nitrogenous bases. And those bonds were hydrogen bonds, so remember that. Now, in a protein, these bonds are peptide bonds. So holding amino acids together are peptide bonds. Please do not refer to these as polypeptide bonds. There is no such thing as a polypeptide bond. It is only peptide bonds between amino acids and hydrogen bonds between nitrogenous bases. So proteins will differ from each other by the type and sequence of amino acids. So in our little scenario, we are saying it will differ based on what shapes are present and the order in which they occur. Because you can get different proteins where you have many different amino acids, and a protein can have all, none, some, various differing amounts of a certain amino acid, and that's what makes them different. But you can also get 2 proteins that have exactly the same types of amino acids in them, but the order in which they are occurring is completely different, and that's what also contributes to a difference in proteins. So let's give this a little bit of a visualization. So let's say that this chain of 4 amino acids, in this order, represents the protein for straight hair. So if you have this sequence of amino acids, you are going to have straight hair. But if I change just one amino acid, and now I have square, square, triangle, circle, I am now coding for curly hair. So I only changed one amino acid. I'm still coding for hair. This is still coding for the structure that is hair. The only thing that I have changed with the one amino acid is I've gone from straight to curly. So there's a different phenotype that's occurring. So you can see that it's very important that we have a very strict set of guidelines or instructions to tell us this is the order that your amino acids need to be in. Because the difference between straight hair and curly hair is like fairly innocuous. It doesn't really make a difference. But it could be, if I change one amino acid, the difference between functioning and non-functioning, which is then really a serious problem. So we need to make sure that we have that set of instructions. Now, the way that I want you to try and think about this is, think about, you want to be a chef. You want to be a cook. You want to go and make this meal that your friend has told you about. They said it's really delicious, that you super have to try it, but you don't know how to make it. So where are you going to go look to find out, how am I going to make this recipe, or how am I going to make this bit of food? What are you going to look for? Besides going and looking on the internet, you're going to look for a recipe. You're going to look for a set of instructions to tell you, this is the ingredients that I need, and this is the steps that I need to do in order to create whatever this meal is. So in our bodies, where do we find the recipes? DNA is where we find the recipes. So the DNA, the sequence of the nucleotides in the DNA, represents the codes and the instructions to create our meals, inverted commas, which is basically our proteins. So in DNA, for every 3 nucleotides, so every 3 individual nucleotides of DNA code, will represent or code for 1 amino acid. So when I put 3 nucleotides together, it will give me, eventually through the process, 1 amino acid. So for example, if I have thymine, guanine, guanine, in my little case example, I'm going to say that this codes for the circle shape, or in this instance, the circle amino acid. It could be another set of letters, it could be another set of shapes. This is just a little example for you to have. So if 3 nucleotides give 1 amino acid, let me ask you this. If I want to create a chain of 50 amino acids for this one protein, how many nucleotides am I going to need? Well, I'm going to have 150 nucleotides. How did I get there? Well, if 1 amino acid needs 3, then 50 is going to need 50 times 3, which is 150 nucleotides. So, just remember that. So, for our little practical example, I'm going to say that thymine, adenine, cytosine will code for a circle. Adenine, adenine, adenine will code for a triangle. Adenine, guanine, cytosine, an octagon. And guanine, guanine, thymine, a square. So this is just a representative example, so we can have an illustration. So if I give you this amino acid sequence, triangle, square, square, what is going to be the representative DNA code for this? Well, in this case, it's fairly easy. We just match like with like. So it will be triangle, AAA, square, GGT, and the same again, GGT, because I am coding for the same amino acid. Here's another example. What is this DNA code sequence going to be? Again, I can do the same thing. I can just read off where I'm looking from. It would be TAC for the circle. It would be AGC for the octagon. And it would be GGT for the square. So that really is protein synthesis made a little bit easy. But what I've actually done here is I've gone the wrong way around. I've gone backwards. So I started with the amino acids and I went back to the DNA. But that's not the way that it works. That's not the way that we have to understand that it works. We have to understand it that we are going to start with the DNA and eventually end up with our protein, our fully formed protein. So now let's get a little bit more into what actually happens in this process. So like I said, we are going to start with the DNA. Here's my 2 polynucleotide chains connected with each other. This is my DNA sequence. So step 1, I'm going to go from DNA and I'm going to create a strand of messenger RNA, mRNA. Remember the differences between DNA and RNA? Go back to the video, make sure you understand if you haven't. I know this does look a little bit funky at the moment, but in the next video we'll go through a little bit more in detail as to why it is a bit funky. But this is basically step 1, DNA to mRNA. And then step 2 is going to be from mRNA to protein. So that's going to be step 2, creating that final protein. So this is the generic overview of what protein synthesis is. We can go a little bit further. We can say step 1, DNA to mRNA is known as transcription, the creation of a complementary mRNA strand from a section of DNA code. So there's that word again, complementary. I'm going to go a bit more in depth in that in the next video, but basically I'm going from DNA to mRNA. That is transcription. Then if I look at step 2, I'm going from mRNA to protein, and that is known as translation, the creation of a protein using the code of the mRNA to join amino acids in a set sequence. And the set sequence is determined by the code on the mRNA strand. So again, step 1, transcription, DNA to mRNA. Step 2, translation, mRNA to protein. So let's look at another way that we can give this a bit of an overview. Here's a visual representation. This is my nucleus. So the green thing is my nuclear membrane going around, and here's my strand of DNA. So step 1 is transcription, where a section of DNA is copied into messenger RNA, mRNA, in a complementary fashion. So we're creating this mRNA strand based on a certain sequence of DNA. Now, it's not the whole DNA that I create into an mRNA. I only need to use the short section that I want to create the protein from. I'm only interested in this section for this protein. So I'm going to make the mRNA strand from just that section alone. Then what's going to happen is the mRNA is then carried outside of the nucleus into the cytoplasm and attaches to a ribosome, this little red thing here. So the mRNA is going to leave into cytoplasm and attach to the ribosome being where the point where we are making proteins. At the same time, we have another RNA molecule, tRNA, this little green thing here, collects and joins with one amino acid, so one tRNA to one amino acid, and it joins with it in the cytoplasm. Then we can move on to our step 2, which is translation. The tRNA brings the amino acid to the ribosome in order to form protein based on the sequence of the mRNA strand. Now bear in mind, as an overview for next video, this is one tRNA and one amino acid. My chain is many amino acids. So this process is going to be happening multiple times. Multiple tRNAs are going to be coming in with more amino acids to create this chain based on the code that is here. So that is really our basic overview of what protein synthesis is. I hope you've got a much better understanding before we then move into part 2, where we're going to really start looking at the in-depth, what I need to know, how do I go from DNA to mRNA, how do I go from mRNA to a protein, how am I going to know which amino acids are going to work, till I eventually get to a final protein. So please go back, rewind, pause, write down, make notes, flow charts, tables, whatever you want to do. Make sure you have a really good basis of understanding for this protein synthesis before we move forward. As always, you can either get me on my Skype for a little meeting, or you get me on Teams where you can ask me any questions that you want. Otherwise, I hope you enjoyed, and I hope that your studying is going really well and that you are hopefully understanding all of this. Thanks very much, guys. Enjoy!